my favorite tree. So it is kind of amazing how a plant can take energy from the sun and make a sugar out of it, because you can't just microwave a plant and have it make sugar. You can't X-ray a plant and have it make sugar. You can't radio wave a plant and make sugar, but you can have sunlight, which is also part of the electromagnetic spectrum, strike this thing and have it make sugar. And that's pretty amazing to me. And I hope to you too. Okay, big picture time for photosynthesis. Let's see how the light reactions in the Calvin cycle are connected to each other. The light reactions happen in the thylakoid membrane of the chloroplast, and that's where sunlight, visible spectrum of sunlight, will be absorbed. Water is a key player here too, because it's the source, endless source of electrons. We will release oxygen, but that's not the point of the light reactions. The point is actually right here, to make ATP and NADPH. Those two molecules are energy carriers that are then going to bring that energy to the stroma where the Calvin cycle takes place. The Calvin cycle is all about taking carbon dioxide and, with the help of an enzyme called rubisco, making these molecules called G3Ps. Now each G3P only has 3 carbons in it, so it's going to take 2 of those to make a 6-carbon glucose. Now that the energy has been dumped into G3Ps, we can send these less energized molecules, NADP plus and ADP, right on back to the thylakoid membrane, where sunlight can be absorbed again to get them all charged up so they can continue their little circuit back again. And that is the overview of photosynthesis. The overall purpose of the light reactions of photosynthesis is to make ATP and NADPH. And those two molecules are then sent to the Calvin cycle in the stroma, where the energy is then dumped into carbon intermediates. So as a reminder, ATP has 3 phosphates. There's 1, 2, and a third, ATP. And ADP has 2 phosphates, 1 and 2. So we're actually going this way right now. We're making this stuff. And so I'm going to have these little cards that say ADP and a free inorganic phosphate. And when we flip it over, that means we've made ATP. Similarly, in this model, there are NADP plus cards. Structurally, the difference between NADP plus and NADPH, I've highlighted them here. You'll see NADP plus just has this hydrogen, and then there's a plus sign on the nitrogen. And then here is NADPH. There's going to be an extra hydrogen added here, and the plus sign is gone. So it's going to be called NADPH. The reality is 2 electrons are involved and that hydrogen, but for the model, I'm just going to represent electrons, plural, with 1 electron. And when we make NADPH, it's going to go into the little pocket, and it's going to become NADPH. And those 2 molecules will go to the Calvin cycle. As a visible light story, photosynthesis is also an electromagnetic spectrum story. As you can see, visible light is that small slice out of the larger spectrum. Red and blue light. Red, we'll say 680 or 700 nanometer. The chlorophyll A reaction center electron is excited by that for sure. Blue light also excites the chlorophyll A reaction center electron, and we'll talk about how both of those are useful soon. Yellow light is not absorbed directly by the chlorophyll A reaction center. However, the antenna complex is going to make great use of this wavelength and many others that aren't exactly the trigger wavelengths. Green is not absorbed so well by chlorophyll A, and it's mostly reflected. Hence, a lot of things that are plant-like look green. Both red and blue photons are able to excite the electron found in the chlorophyll A reaction center. The energy associated with red, or wavelength around 680 nanometer, causes that electron to be boosted from ground state to an excited state, where it then becomes captured by pheophytin, which is the name of the primary electron acceptor associated with photosystem 2. And then very quickly through redox, that electron becomes part of triangle structure, and then it moves this way through redox. But what about blue light? Blue light, more energy, say like 450 nanometer, also excites this electron. It's tuned to it, and it will go past the first state and go to another higher excited state, where it quickly drops back down to the original excited state and becomes part of the structure of pheophytin. And then same thing through redox, that electron is transferred to triangle and then off the page. So red and blue light can excite the electron. However, there's more to light than red and blue. For example, there's yellow. Now, in the case of yellow, the electron will not jump for yellow. But the antenna complex, also known as the light harvesting complex that surrounds this, there are pigments there that are able to absorb the energy of yellow. But when they do that, it causes vibrational energy, and that vibrational energy is then transferred through adjacent pigments that are tuned to that energy, and the net result of this is a loss of energy as it travels through this path. And once it has lost enough energy, it will then reach the energy of this, because that's sort of the lower energy side of things at 680. And so the equivalent energy packet associated with this photon is delivered, and that will cause this electron to jump anyway. So we'll jump here, become captured by phaophyton, then through redox, it'll jump onto triangle, and then move to the right. Okay, time for the light reaction. So we're going to start off with red light at photosystem 2, 680 nanometer, at photosystem 1, 700 nanometer wavelength. At the same time, those electrons are going to get excited, boosted from an energy level, and then quickly bonding to primary electron acceptors here and here. We're going to come back to this one a little bit later. Let's focus on photosystem 2. So now that that electron is not here, let's follow its route. So we'll have a little redox moment. This is a laterally diffusing protein, and it's going to bring it over here and pass it off to dinosaur egg. So that will lead to active transport, translocation of a proton to the thylakoid interior. Then we'll redox this thing again, lateral transdiffusible protein through the membrane, and it's going to take the space of that one which left earlier. But let's go back over here again. Clearly there's a problem, no electron here. We can solve that. Boom, solved. This thing is called the water oxidizing clock. It's basically a little storage place for electrons, but oh no, it's empty. We can solve that. Boom, 2 water molecules just split. So this is now going to come together, an O and an O makes, you guessed it, O2. That's diatomic oxygen. That's the stuff we breathe. We're happy about that. The 4 electrons will fill the clock. Problem solved. 4 protons are going to be released from those 2 waters too, and they're going to join the other proton from dinosaur egg, and they're going to go way over here to ATP synthase, where we're going to couple the exergonic motion of diffusion to the endergonic work of making ATP. So boom, ATP is made. But we're not done yet. We're going to go over here and continue with the fate of this electron. So this one is going to become part of the structure of star. Star is going to move over to strange shape, which is really called NADP reductase. So strange shape here, that electron, along with a proton, which I'm not showing, will become part of the structure of NADPH. Now, ATP and NADPH, made in the light reactions, they're going to go somewhere else. Follow me. And look where they have gone. They have gone here, and this is called the Calvin cycle, and this is where we're going to make some sugars in another video. In non-cyclic photophosphorylation, electrons are on a one-way trip to the structure of NADPH. So when one photon excites an electron and it goes through its routine, we're missing one here. It doesn't come back. So we need to replace it from somewhere, and that's what this water oxidizing clock is all about. But right now, that's an empty clock. So how do we fill that clock? That is when 2 water molecules are split to take 2 oxygens to make O2, diatomic oxygen. That's the stuff we breathe. The clock is then filled with 4 electrons. That's the most that it can hold. These 4 protons then contribute to the H+ gradient on the inside of the lumen, and that's connected to making ATP, forming ATP later. So let's do this again. We strike the electron, it boosts it, primary electron acceptor, electron transport, gone. The clock replaces the missing electron. Here comes another photon, photon number 2. Excited, gone. We replace the electron. Photon number 3. Excited, gone. We replace the electron. Photon number 4. Excited, gone. We replace the electron. So That's 4 photons. We emptied the clock, and at that point, you'd break open 2 water molecules and fill up the clock again. So every 4 photons, I guess, technically, you make oxygen.